Chapter 1, What is Life? Biology is the study of life, taken from the suffix bio, which is Latin for life, and ology, which means the study of. Put them together and you have biology, the study of life. Here's a list of the primary objectives for this chapter. Each chapter will have a list of these objectives on the second slide. Most of them are self-explanatory, but in the case of these objectives, I'm going to go ahead and read them to you because they need some more explanation. So the first objective is to list and define the 5 basic characteristics of life. I know the book says there's 9, but we're really just going to focus on 5 basic characteristics. Determine the 3 parts of the cell theory. There's really 5 parts of the cell theory, but just to keep things simple, we're going to say there's 3 for this chapter. Differentiate between the 3 domains and 5 kingdoms of organisms. We're going to learn the scientific name for humans and know what domain and kingdom humans belong to. We're going to arrange the levels of nature from simplest to most complex, and we're going to list and define the components of the scientific method. Here are the 9 characteristics of life from your book. A living thing is capable of responding to its external environment or external stimuli. Living things are capable of altering the environment. Living things sense the environment. Living things are capable of adapting to the environment. Living things maintain a constant internal environment. The maintenance of this environment is what we call homeostasis, and we'll talk about it further in these next few slides. Living things capable of using energy. Living things contain materials only found in living organisms. Living things have a high degree of organization. And finally, living things have the ability to reproduce and ensure that their species continues to exist. Here are the pictorial representations of the 9 characteristics of life from your book. Like I said, living things are capable of responding to external stimuli. Living things alter their environment. And for this one, a good thing to think about would be like humans building homes or birds building nests or something like that. Living things are capable of sensing the environment so they can respond to it appropriately. And we're going to spend a whole chapter talking about the organs that organisms use to sense their environment. Living things adapt to the environment. Living things use energy, which they either gain from the sun's light if they're a plant, or they take from the chemical bonds contained within the foods they eat if they're an animal or any other organism that eats. Living things are capable of reproducing, which is actually the ultimate goal of any organism. Living things contain certain materials, mostly carbon, and also hydrogen, nitrogen, oxygen, and a few others that we'll talk about later. Living things maintain a constant internal environment, which is called homeostasis, which we're going to talk about in great detail. You may not be real familiar with what homeostasis is right now, but by the time we're done with this class, you're never going to get a question wrong about homeostasis. And living things display a high degree of organization. Here are the 5 basic characteristics of life that we're going to focus on in this chapter. And they're very similar to the 9 characteristics of life in your book. But you can forget about some of those because they're sort of reiterated here, so I guess you don't really need to forget them. You just need to think about them as these 5. Living things are capable of taking in energy and material. Living things are capable of sensing and responding to changes in their environment. And a response would be something like, it could result in adaptation or the living organism altering its environment. Living things consist of one or more cells, and we'll talk about more what a cell is later and what defines a cell. Living things are capable of maintaining homeostasis or a constant internal environment, and living things are also capable of growing, and that's doesn't really matter how many cells are contained within that living organism. Like a single celled organism still grows, and all living organisms, or at least ones that are most definitely defined as a living organism, are capable of reproducing. The first characteristic of living organisms that we're going to talk about is that living organisms are capable of taking in energy and material. And this energy always comes from their external environment, either from the light of the sun or from the bodies of other organisms. The maintenance of the organization of any organism requires this energy. And there are three categories that organisms are divided into based on how they obtain their energy. The first one is producers, and they make their own food using energy from the sun and abiotic nutrients from the soil. Consumers include both herbivores and carnivores and also omnivores, and they derive their energy from the organisms they eat. And they eat both producers, which are plants, or other consumers. So just remember that a cow is not a producer, it is a herbivore, which eats plants and is therefore a consumer, as are carnivores and omnivores. And the third category are the decomposers, and they derive their energy from dead organisms. So these would be something like fungi or anything that makes other organisms decompose. So like fungi and bacteria. You can also divide organisms based on the food they must eat to gain this energy. So herbivores only eat plants, carnivores only eat animals, and omnivores eat both plants and animals. And of course, humans are omnivores because we eat both of those things. And the food can be broken down into nutrients, which can be further broken down into molecules, and then the individual bonds within these molecules can be broken down for energy, or the molecules can be rearranged to make new molecules, and the energy required to do work results in an organism being capable of moving or motion. And here's just a picture of the methods by which organisms take in energy and food. For plants, this process is called photosynthesis, and they take in light energy from the sun and CO2 from the air to make glucose, which is their food source. For organisms that don't carry out photosynthesis, they carry out respiration, in which they take in oxygen from the environment and get energy from the food they consume, and all this results in the production of water and CO2. And I guess I should remember to point out that in photosynthesis, the plants release O2 and take in CO2, and we of course do the opposite. Here's a diagram which displays the methods by which producers, consumers, and decomposers take in nutrients and energy. Producers take in their energy from the light of the sun, and they gain their nutrients from non-living materials, which in the case of plants would be the soil that they're planted in. Consumers, of course, eat other consumers and or producers, and that's where they obtain both their energy and their nutrients. And decomposers are essentially the same way. They obtain their nutrients from the bodies or material that they break down, and they also obtain their energy from this process also. And I'd also like to point out that at all levels except when producers take in nutrients and energy from the sun, heat will be given off as a waste product. And here's a picture that displays metabolism. And metabolism we'll define here as the process by which an organism either breaks down or builds up its own nutrients. So this can be divided into two parts. Catabolism is where these materials are broke down. So they break the larger molecules into smaller molecules in order to obtain energy from the chemical bonds of those larger molecules. And in anabolism, they build these small molecules back up into larger ones, which takes energy, but it allows them to create the molecules they need to maintain themselves. The second characteristic of life that we're focusing on is that living organisms sense and respond to changes in their external environment. And we're really not going to get too deep into this because I feel as though it's pretty self-explanatory. So, organisms sense and respond to internal and external stimuli, and they use this information to respond appropriately to the external environment. They can either adapt to it or alter it. Living organisms also consist of one or more cells. Those organisms that consist of one cell are called unicellular, and these include bacteria, archaea, protists, and fungi. And archaea are a whole lot like bacteria. They generally just live in more extreme environments. Protists are single-celled eukaryotes, and we'll talk about what a eukaryote is here in a few more slides. And fungi can also occur in one cell. And the organisms that have more than one cell are called multicellular, and these include some protists, fungi, plants, and animals, of course. Here are the three parts of the cell theory that we're going to talk about in this chapter. All organisms consist of one or more cells, like I said in the last slide. The cell is the basic unit or building block of life. You can't have a living organism without a cell, obviously. And all cells must come from pre-existing cells. They can't just appear out of thin air, basically. Living organisms also maintain homeostasis, and here we're going to finally talk about homeostasis in greater detail. And homeostasis is the maintenance of a relatively constant internal environment. So you maintain a constant body temperature, 36.5 to 37.5 degrees Celsius, or 98.6 degrees Fahrenheit. And this level of maintenance depends on the external environment, like how hot or cold it is, or what kind of stressors you might be exposed to, or whatever else. All body systems in humans work to maintain homeostasis, and this is both chemical and physical homeostasis. Let's break down this word homeostasis. Homeo means unchanging in Latin, and stasis means standing in Latin. Therefore, when we put these together, homeostasis means staying the same. Humans and other organisms only function properly within narrow ranges of temperature and chemistry. An example of maintaining homeostatic chemistry would be to maintain a constant pH, which for humans and most other organisms is about 7. And this ensures that the body's internal environment maintains relatively constant and within physiological limits. And homeostasis can be maintained by both conscious and unconscious controls. So an example of this would be if we experience a reduction in our body temperature, the blood vessels would constrict or get smaller in response to this lower temperature, and we would also consciously seek out more and warmer clothing. We regulate homeostasis with feedback loops, and a feedback loop is just the means of receiving and responding to information, and these can be either positive or negative, and they come with three components. First, they all have receptors, which they use to monitor the environment and report any changes. So these send input signals, which can either be nerve impulses or chemical signals, that eventually are received in the brain. And the brain would be our control center, which is the second component, and this receives and evaluates the receptor signals and formulates a response. And lastly, we have the effectors, which receive the signal from the central control center and carry out a response. So this would be sort of like a reflex, except that we would receive the information with the receptor, which would only go to our spinal cord and just loop straight to the effectors, so we can respond with a knee jerk or any other kind of reflex. Here are the two types of feedback loops. The most common is the negative feedback loop, which works to reduce, reverse, or eliminate changes detected by the receptor. So an example of this would be when you feel as though you're the wrong temperature. Say you're too hot, your body tells you you're too hot, you feel bad, and then you either move to a cooler area or a shade, or maybe you drink water or something like that. In comparison, positive feedback loops are pretty uncommon, and they work to increase or reinforce changes detected by the receptor. And two examples of this would be urinating or giving birth. And over here we have the diagram which displays this. So up here we have some stimulus that disrupts homeostasis and increases or decreases the controlled condition. The receptors receive this information from the external environment, and they usually send it as a nerve impulse to the brain, which is the control center, and this formulates the response, and all in the form of sending a nerve impulse to the effector, and they carry out the response from the control center, and the response alters the condition. And like I said, the negative feedback loop negates it, while the positive feedback loop enhances it. The fifth and final characteristic of life that we're going to cover is that living organisms reproduce and grow. Reproduction, from a purely biological perspective, is the ultimate goal of an individual organism's survival. Everything that they've done up until this point has been so that they can survive and reproduce to continue on with the species to help maintain that species. And this holds true for cells as well. All cells must come from pre-existing cells. All organisms must come from other pre-existing organisms. And life must come from other pre-existing life. And this, of course, allows for the continuation of species, like I said. Organisms can only reproduce with other organisms of the same species to create viable offspring. Obviously, there's some hybrids out there where two animals or organisms of different species can reproduce to create these hybrids, but such hybrids usually are sterile, such as mules and zonkies are nearly always sterile, at least the males are. For animals like ligers, this holds true over most cases also. In addition to being capable of reproducing, living organisms also grow and develop, and we're going to find growth here as an increase in the size of an individual organism or in the number of cells within that organism. And I'd also like to point out that even in single-celled organisms, the individual cells still grow larger. And we can differentiate this between development, which we'll define as the maturation of an individual organism, which includes repair after injury. And here below is a very basic display of development. And this shows an embryo develops into a fetus, and this fetus will be born, and then we'll call it a child. Children grow into adolescence, and adolescents become adults. Let's talk about cell reproduction, and there are two methods by which cells reproduce. The first one is mitosis, which is the process that our somatic or regular body cells carry out to make more of themselves. And this results into identical daughter cells, which contain the same amount of genetic material as their predecessor cell. And then there's meiosis, which is what our germ cells carry out to create gametes, which are eggs or sperm. And this creates two haploid daughter cells. Haploid means that these cells contain half of the amount of genetic information as their predecessor cells. And of course, all cells contain DNA, which is the molecule which contains genes and ensures heredity. The gene is the basic unit of heredity. This DNA is duplicated in daughter cells regardless if they contain half or all or the same amount of genetic information as the predecessor cell. And DNA is, of course, arranged in what we call an antiparallel double helix strand. And we'll talk more about DNA, at least in the next chapter anyway. And this is, both of these cell reproductive processes are necessary for the reproduction of organisms. The male gamete or sperm combines with the female gamete, what we call the egg or ova, and this creates a single-celled zygote, which, since it contains half the genetic material from the male and half the genetic material from the female, it contains a full set of genes. Moving on to the second section of Chapter 1, Human Biology is Structured and Logical. The natural world, of course, seems overwhelming and chaotic at first, until we organize it, which is, of course, the reason why we have organization in the first place. Biology is organized into steps from the microscopic to the macroscopic levels, and I should have just said level for all of this. Basically, small units make up larger units, which in turn form still larger units. And we'll talk about this organization in greater detail in a second. And of course, we have an artificial classification system, or taxonomy. We use these names to identify organisms and to show relationships between different species of organisms. And we group organisms based on similar characteristics or similarities in their DNA, which is different from physical characteristics, of course. Now we're going to talk about the different classifications of living organisms. And as this is a primary objective, you know you're definitely going to be tested over it, so pay attention. There are three domains of life: the eubacteria, which includes prokaryotes and other unicellular organisms. And then there's the archaebacteria, which are essentially the same as eubacteria. They're genetically different and live in extreme environments. And many of them are what we call extremophiles because they live in such extreme environments, like very hot environments or very acidic, salty, what have you. And then there's the eukarya, which are the eukaryotes, which consists of both unicellular and multicellular organisms. Guess which domain we're in? There are 6 kingdoms of life. There's Animalia, which includes eukaryotes, which are, of course, multicellular. Then there's Plantae, which also includes eukaryotic multicellular organisms. And then there's fungi, which are always eukaryotic, but they can be multicellular or unicellular. And then there's Protista, and they're all eukaryotic and can also be multicellular and unicellular. And I guess I better give you an example of a protist, which would be an amoeba is a protist. And then there's the eubacteria, which are all prokaryotic and unicellular and include bacteria. Eubacteria actually just means true bacteria. Eu means true. And then there's the archaebacteria, which are pretty much the same as the eubacteria. Here's a diagram which shows the domains and kingdoms of life. There is, of course, the eubacteria, the archaebacteria, and the eukarya. The eubacteria contains only one kingdom, the bacteria. The archaebacteria also contains one kingdom, the archaea, which are just bacteria that live in extreme conditions and are called extremophiles. And then the eukarya contains 4 kingdoms, the protista, things like protozoans and algae, the animalia, which we're in, of course, the fungi, and kingdom Plantae, which includes the plants, of course. Here's the classification scheme for the human species as compared to the house cat. We're both in domain Eukarya, we're both in kingdom Animalia, we're both in phylum Chordata, which includes all animals that have a definable backbone. We're both mammals, so we're in the class Mammalia. We're in different orders though. Humans are primates, house cats are carnivora. Different families, of course. Humans are in Hominidae and cats are in Felidae. And of course the genus and species for humans is Homo sapien, and the genus and species for the house cat is Felis domesticus. And we're going to say here that species implies reproductive isolation, which is members of a particular species must be able to produce viable and fertile offspring only if they breed with other members of the same species, and there's very, very few exceptions. Like I said, there's some hybrids that can reproduce, but there aren't many of these anyway. And here's just some pictures that reiterates what I just said. We're in the kingdom Animalia, which contains all multicellular organisms that ingest nutrients. We're in phylum Vertebrata or Chordata, like I said in the slide ago. And I shouldn't have said that it's all animals that have a spinal cord. It really should be called a dorsal hollow nodal cord. All of them do look, of course, like spinal cords. We belong to the class Mammalia, the order primates, family Homidae, genus Homo, species Homo sapien. Now we're going to talk about life's levels of complexity, and here we have a diagram of the organization of life from the simplest to the most complex. The simplest elements are abiotic, and the first one is the atom, which is the smallest unit of an element, which is preceded by or followed by the molecule, which contains 2 or more atoms. And then we move on to the biotic, the cell, which is the smallest structure of life. Groups of cells create tissues, and these groups of cells then combine to form organs, which contain 1 or more types of tissue, at least 2 types of tissue anyway, and they function together. A bunch of organs together create an organ system, and then when we put a bunch of organ systems together, we of course get a whole organism. And then a group of organisms of the same species is a population. A group of several organisms from different species is a community. All of these things together all these groups of organisms create an ecosystem, and all these ecosystems together create the biosphere. Don't worry too much about these four on the bottom, but just know they're there. So here's just a picture of what I just said. The first level of organization is at the chemical level, which is made up of atoms, which make up molecules. In this case, it's a DNA molecule. These molecules combine to create cells. Of course, it's a little bit more complicated than that, but we'll just say they combine to create cells. A bunch of cells together creates a tissue, and then a bunch of tissues together creates an organ. Groups of organs combine to form organ systems, and of course, at the organism level, we are made up of all of these organ systems. Moving on to section 3 of chapter 1, using science to learn about the natural world. Here we're going to define science as a way of thinking about the natural world. A scientist is then a person which makes and tests predictions or hypotheses to support or disprove a proposed explanation. Science and scientists should be objective or have some sort of end goal in mind, is what I mean here. There are, of course, some limitations. Scientific conclusions may change or be modified as our understanding and technology increases, of course. Science is a systematic study of nature. In order to make scientific discoveries and breakthroughs, we use the scientific method, which consists of 6 steps. The first step is that we must make an observation about the natural world, and we use these observations to create a hypothesis, which is an educated prediction of what we saw. We then need to perform experiments to test this hypothesis, and likely you'll need to repeat the test multiple times or perform multiple new ones as you learn through doing more about your hypothesis or what you observed. We then collect and analyze the data from these experiments, and then we report the test results and make a conclusion. We ask ourselves, does the data confirm or reject the hypothesis? And then scientists will repeat the experiment, either themselves or maybe another group of scientists repeats the same experiment, just to see if the hypothesis is still supported, and if the hypothesis is tested many times, this could possibly result in a scientific law. And there aren't really many scientific laws in biology, just to clear that up real quick. Here's a diagram which displays what I just said, but probably in a less confusing way. Step 1 is to observe, where we recognize the problem or the unanswered question. We then create a hypothesis based on what we've observed, and this is developed to explain the problem. We make predictions based on the hypothesis and then perform experiments. And this step involves both the design and actual performing of the experiment to test the hypothesis. We collect and analyze the data from these experiments and interpret this data to reach conclusions. We ask ourselves if the hypothesis actually predicts reality. If no, that means we must form a new hypothesis and perform a lot of the process over again. If yes, then we just keep experimenting to further confirm the hypothesis. And after this is all done, a scientist will communicate the results to other scientists, and this new knowledge results in new questions. So basically you end up with new groups of scientists performing the same experiments over and over until they confirm that the hypothesis is a law, which of course doesn't really happen all that much. And then over to You're right is an example of a real simple experiment that I really wouldn't waste too much time on. Just know what the scientific method is and know what we'll say is 6 steps so long as you count repeats. Here are the components of a good study. First, any good study has to have a control group, which is the group to which the experimental group can be compared. And this is the group where you basically do nothing to it. In like a human drug study, this would be the group which is exposed to the placebo pill, which is a pill that does not contain the active ingredient. So essentially we've done nothing to this group. And then there's the experimental group, which is actually exposed to the experimental variable, and then compared to the control group to see if there's any difference. And a variable is, of course, a factor that can change with different circumstances or time. So, like in a drug test, this would be like the different drugs they test. And specifically, an experimental variable is the factor being tested. Let's talk more about what a scientific theory is. And a theory explains a large number of observations. Know that this is different from a law. A theory isn't confirmed as much as a law, which takes lots and lots of tests to be confirmed as a law. But there's some common theories out there, obviously. There's the gravitational theory, the cell theory, which is the main theory we're going to talk about over the next few chapters. The germ theory, what causes illness. The plate tectonics theory, evolution, and of course natural selection. Just know there's a difference between the word theory and law. Theories are less proven, they can be disproven. Laws have been proven to death, they're not going to be disproven. That's the basic difference. There are essentially 2 things that make a good theory. First, it must be supported by tens of thousands of experiments, which, like I said, requires a lot of experimentation to be developed. So, even though a theory takes tens of thousands of experiments to be proven or supported, a law takes even more. And of course, a theory may be modified or even rejected upon further investigation, which of course further separates a theory from a law. In order to properly carry out the scientific method, we must use critical reasoning. And there's 2 different types of critical reasoning. There's deductive reasoning, also known as top-down logic, which you draw a conclusion from multiple premises. And this is in the form of an if-then statement, like if I do this, this results. Sort of just cause and effect. And this is actually used to design an experiment using the scientific method. Then there's inductive reasoning, which is bottom-up logic, which involves multiple premises supplying strong evidence for the conclusion, and this is actually used to develop hypotheses. And basically for these, you just need to know that deductive reasoning is used to design an experiment and is an if-then statement, and inductive reasoning is used to develop a hypothesis. And like I said, critical reasoning is most definitely useful in science. It's used to analyze the data and analyze the experiments and the claims before accepting what you read. And a good scientist needs to be skeptical, he needs to be logical, and he needs to be open to new information as it comes. Because you need to understand that information changes and new information presents itself. This slide pretty much has just really good advice on evaluating scientific information. We need to let sound scientific evidence or well-supported scientific evidence, not opinions or speculation, do the convincing. You need to evaluate the source, question the motives, who's funding this research, why are they doing it, what's their motivation, and all of those things will probably determine in what way they're biased. And question the credentials. You don't just want to listen to anybody that's not a scientist or doesn't perform their experiments well. And you also need to evaluate the content. Is it made up of facts, which are all true things that are said? Is it made of opinions, or just what people think? They're neither really right or wrong, I suppose, but you just need to be careful. And is it speculation? You don't want to listen to just speculative facts that can be either true or not. We just want the facts which can be proven true or not. Here's just some tips about reading scientific information. Most real good scientific information is actually in scientific journals. And these, like I said, are the best source of information. Yeah, they're difficult for the layperson to understand, but they still are the best source of information. One of these reasons is that they're peer-reviewed. Peer scientists determine the merit and quality of the work, and you could differentiate this between not peer-evaluated works, such as those found in magazines, which just include opinions. And then there's secondary sources, which are of course not the best source, but the layperson can understand them. These are contained within the news, the magazines, internet, other media. Be very careful of information on the internet. A lot of the information gathered there really just depends on how you type a sentence into Google or something, so be careful. And you use reliable sources, such as URLs of edu, .gov, .org. These days these are becoming somewhat questionable themselves. For the most part, you can trust edu, .gov, maybe not .org so much anymore, but what can you do? Be careful about what you read. Here's a table taken from Chapter 2. It's really going to be the only thing we study in Chapter 2. There might be a few things in the evolution slides at the end, but we'll just kind of cross that bridge when we get to it. And these are just the organ systems of the body and their function. You can read the functions for yourselves. I'm just going to talk about which ones we're covering and which ones we're not covering. We are not going to cover the skeletal-muscular system. We'll cover the nervous system, the sensory system. We'll sort of cover the cutaneous system or the skin. It's kind of in the lymphatic system as it's a barrier to microbes, or I guess I should say the immune system. Then we're going to study the lymphatic system or the immune system, the cardiovascular system, the respiratory system, the digestive system, the urinary system. We're not going to study the endocrine system. We'll just kind of address it within the other systems when we get there. And we'll also cover the reproductive system. And this was my first set of slides. I hope I did okay, and I hope you learned something from them. Have a nice day.